everybody. I'm glad to see those early arrivers, and I guess that the third day in a conference might get some people to oversleep in the morning, or they might be deterred by the subject, because I'm going to speak, as my abstract and my previous talk have been focused on theoretical approaches. My lab is a theoretical lab. My background is theoretical chemistry, chemistry and physics, quantum chemistry, and I moved to chemoinformatics and drug discovery in the more recent years. And I'd like to talk to you today about some problems that might have implications to delivery systems, although I'm not going to talk at all about delivery methods or delivery technologies today. So discovering drug candidates in silico has encountered quite a few problems in recent years, and I'm going to present some of those problems and hopefully convince you. But you can see already on my cover slide that the main idea that we're developing in my lab today is multi-targeting of molecules. So we're talking about single molecules that can hit several receptors simultaneously, not the same molecule hitting at the same time all the receptors, but of course different, the same molecule, but since it is in some concentration, some of it would hit receptor C and some of it would hit some of the other receptors. And one of the examples is this molecule that hits both acetylcholinesterase and beta secretase 1 and butyryl acetylcholinesterase and beta amyloid aggregates, and is a candidate for curing Alzheimer and for studying effects of Alzheimer in small animals. So I'm going to start with problems in current drug discovery, and one of the main problems is that many disease conditions are multifactorial. They have many targets. Angiogenesis is one of the most well-known ones, and some of you who are involved in issues of cancer and angiogenesis know well that all those receptors and others are involved in one way or another in angiogenesis. And so hitting or blocking one of them, blocking just the EGF receptor, might not be enough, might not be sufficient in order to treat, to block angiogenesis in cases of cancer. Another well-known case is HIV. HIV-1 has already in the clinic has a few different inhibitors that block processes starting, and the processes are named here from the entrance to from the binding to CD4, and then the introduction, the fusion into the T4 cells, and then the reverse transcriptase. There are reverse transcriptase inhibitors, and there are protease inhibitors, and others. So there are many locations where one could block HIV, and currently the standard of treatment is a cocktail of drugs. And here we encounter immediately another problem, because if we want to block, this is HIV protease, this is the famous example of an enzyme, a retroviral enzyme that was crystallized in 1990, and 5 or 6 years later, there were already, due to the crystal structure, there were already 4 or 5 drugs on the market from different companies based on the crystal structure that came from the group of Alexander Vlodauer at the National Cancer Institute in Washington. But HIV protease is an aspartic proteinase. It's not the only aspartic proteinase. There are many others aspartic proteinases. There are many other serine proteases. There are many other metalloproteases. And each of those, when we plan to hit one of them, we might hit many of the others. It's very well known with the metalloproteinases. Currently, there is no clinical drug against any of the metalloproteinases, although there are already picomolar IC50 inhibitors of some of them. MMP2 and MMP9 are the most sought after with respect to cancer. So high affinity of a drug might even reduce selectivity rather than increase selectivity, because the strong binding is not only strong binding to one, but is strong binding to a few others that are of the same family. This is a story of the most selective anti-cancer drug that was found some 15 years ago, Gleevec, which was brought into the market and used against chronic myeloid leukemia. The structure is down here, and it was expected to be a direct bullet to hit just the kinase that is ABL in its chimeric form of BCR-ABL, one of the 520 kinases, and it was hoped that it would not hit any other one, but apparently, although the idea was to block the ATP, since the idea is to block the ATP binding site and prevent the transfer of a phosphate to a substrate protein, which is the one that is responsible for the development of chronic myeloid leukemia, of CML. It was found later that that Gleevec has an IC50 of 0.6 micromolar to ABL, but it has even a lower, a better affinity to some of the other kinases such as c-KIT and PDFRs and a few others. So there is no way to go around this, because molecules that hit in high affinity one protein would hit other proteins in either better or lower affinity, but those will be the ones that are responsible for side effects in many disease conditions. So the alternative that has been proposed a few years ago in the mid-90s by some groups, especially some Italian groups, Bolognese, if the name means anything to you, that multitargeting is a better strategy. And if it is, then what is the way to do multitargeting? You can multitarget with a cocktail of drugs. You can multitarget by linking 2 or 3 drugs by a chemical linker, or you could try to design a single molecule that would hit a few targets simultaneously. So I'm quoting here from a paper of 2007 in Drug Discovery Today. The drugs designed to act against individual molecular targets cannot usually combat multigenic diseases such as cancer and many others that affect multiple tissues or cell types such as diabetes, immunoinflammatory, neurodegenerative disorders, and others. Combination drugs that impact multiple targets simultaneously are better at controlling complex disease systems and are less prone to drug resistance and are the standard of care in many important therapeutic areas. So there are already a lot of multi-targeting strategies with disease conditions, but then the problems, the main problems are that there are cocktails in most cases, and those drug combination or combination drugs have other problems such as drug-drug interactions, metabolic interactions, blocking of some of the P450 enzymes that are metabolizing drugs, and then other drugs would stay different. So the pharmacokinetics of the drugs, when given a cocktail, the pharmacokinetics are becoming very complex, and toxicity is one of the results, and of course side effects. Multi-targeting by linking drugs has been tested in a few cases. This is a combination of two drugs that are used in order to reduce symptoms in Alzheimer. The galantamine, which is an acetylcholinesterase inhibitor, and the memantine, that is an MNDA antagonist. So they were linked by a chemical linker, as you see here, just by 4 carbon atoms. And the obvious result is that you have a much larger molecular weight of the molecule, and it also causes problems of solubility because in most cases the lipophilicity is also increased tremendously. So the better solution is to combine combine different drugs into a single molecule, but that has been a very rare event. And I'm aware only of this single success in combining a very well-known acetylcholinesterase inhibitor, which was on the market already since the mid-90s, designed and produced by a colleague of mine in the School of Pharmacy, Marta Rosin. It was sold by Novartis for about $1.5 billion a year. Rasagiline from the group of Musa Yudim at the Technion is a MAO inhibitor, a MAO-B inhibitor, and combining the two was supposed to bring better relief to Alzheimer patients. It has not passed the clinical studies. It failed in the last stages of clinical studies. But the idea of combining two molecules into one and having the function, the acetyl function of rasagiline combined with a carbamate function that is essential for the blocking of the serine of acetylcholinesterase, this was an ingenious chemical idea, and I hope that there will be more, but that's a very difficult idea to achieve in the lab. So just to mention some of the advantages of multi-targeting by single molecules. So we have single molecule pharmacokinetics. We have the possibility of using low affinity binding rather than very high affinity binding, and that increases the number of relevant targets and reduces the side effects and the toxicity. And there's also less resistance due to mutations in cancer, bacterial and viral diseases. So the resistance is also improved. And this is, again, the illustration that I had on my cover slide. Another completely different problem, which was emerged about a year and a half ago when this molecule from Lilly, semajacestat, a gamma secretase inhibitor, which was supposed to be the the best possible drug against Alzheimer and to cure Alzheimer, not only to improve the symptoms as the acetyl-CoA, as the current drugs on the market are doing. It failed. It failed in clinical studies, in phase 3 clinical studies after many patients have been given the drug, and it failed because there were many side effects, and the reason for the side effects was that protein-protein interactions are not single interactions. There are very rare cases where a single protein, this is a map of protein interactions, a single protein, a single interaction of a protein is quite rare. Most of the proteins interact with many other proteins or with many substrates. So the problem is that if we block an enzyme or if we block a protein, we might block not a single interaction that is harmful, but we might block many of its interactions, which are essential for the living system. So the question is how to block out of the interaction of protein A with protein B, protein C, protein D. How could we block just the harmful interaction with protein B and not touch the interaction with protein C or protein D? This general approach has already been given a name, which is substrate selective inhibition. And in some cases, it is possible, such as this is thrombin, which has 2 exosites that interact with different proteins, while the active site is over here. This is where the cleavage of the peptides or proteins takes place. But if we block exosite 1, there is still possibility for other substrates to bind at exosites 2 and vice versa. So this is a relatively easy this is what we call a low-lying fruit in the terms of designing chemical inhibitors. However, there are much more difficult cases, and this is a general explanation of the more difficult cases. So in most proteases, and this is the what is called the Schechter-Berger designation of protein pockets, and this is the peptide that is being cleaved, and the cleavage is between amino acid at position P1 and amino acid in position P1 prime. This is the N-terminal site. This is the C-terminal site. So this is the cleavage site. This represents the enzyme and the enzyme pockets. And the question here is whether we could avoid blocking the cleavage site and still find a way to block substrates on either the N-terminal side or the C-terminal side. And we've chosen, after many studies and literature search, we've chosen this protein that is called prolyl oligopeptidase. It's a serine protease, and it cleaves many, it has many substrates. Among them, we have the LHRH, the luteinizing hormone releasing hormone, the thyrotropin releasing hormone, angiotensins, neurotensins, oxytocin, substance P, vasopressin, and others. So again, you see there are many substrates, and if we wish to block just one of them or just a few of them, this is a difficult case. So what we're trying to do, we decided to pick those 2, angiotensin 3, which is cleaved between P1 and P1 prime. Phenylalanine is the C-terminal of this peptide, and this is the way it binds. This is the way it binds to the enzyme, and thyrotropin releasing hormone TRH is a shorter peptide, and it is cleaved between proline and the amine on the carboxyl on the carboxyl side. So the designed inhibitors have to avoid the P1 prime and only focus on this area between P5 and P2, which are the binding, which have binding pockets on the enzyme. So we've done this with several methods, and the main approach that we use in our lab is this algorithm that I already described on day one, and I'll repeat shortly. The idea of this algorithm, it's a classification algorithm, and the classification, in order to differentiate between molecules that are active and millions of molecules that are not active on a specific target, we would like to find out what is the reason for the difference between those and those. And for classifying molecules, we need to determine on two levels, which I'll illustrate in a minute. The first is which properties distinguish actives from all the others, and then which properties distinguish the best actives from the worst actives. So there are two levels here, and the equivalent of this is if you wish to look in a large population for the best basketball players, you may use height limit and ask everybody who's taller than 2 meters to step forward, and then you will have the what we call the hits, which will be equivalent to micromolar activity, or what is called in chemistry hits. But in order to get leads, we have to understand the difference between the high actives and the low actives, and that is much more difficult, but it allows us to discover nanomolar compounds. So the equivalent of this, continuing this basketball example, if we take the NBA team and have them play against Maccabi Tel Aviv, which is our local best team, NBA would always win against Maccabi. So those would be the high actives and those would be the low actives, although the differences are not in height, the differences are not in color, the differences are not, maybe they are all those are from the same neighborhood even. So the difference here is much more delicate, and it goes to the length of muscles and to the size of the hand and to the ability to jump and so forth. So we have to use many more descriptors or properties of in order to understand the difference between the best ones and the least best ones. So this is what this algorithm does. This is an in-house algorithm called iterative stochastic elimination. It was implemented in order to create classification models, and it won an ACS contest in 2000 for emerging technologies, and developed to do this job over the years. And what ISE does, it constructs the best set of filters. So it's not a single filter, it's lots of filters. It could be 1,000 filters at once that distinguish between two activity classes. And it orders filters by their classification ability, which are later used as weights in order to score molecules. So here are the filters. This is filter number 1, the best one. This is the second best, third best, and so forth. And now we pick a large group of molecules from a chemical database, whether it is a combinatorial chemistry database, completely virtual, or it is a commercial database, which we can buy in the next few days. And that is what we usually do. So we pass those molecules. Molecules that pass more filters get a more positive number. Molecules that do not pass as many filters could get a negative number. It is normalized, so all the numbers are between -1 and +1. And the way for us to represent those molecules is by a group of molecules for which previous results have been known for their activity. We present them in a numerical form. We present their properties. We associate between the properties and the molecular activity by our algorithm and create a model. And once this model has been established, we can pass molecule with unknown activities. Another way to show this is that we start with a database of some 50 actives and many more inactives, because the number of actives must be diluted in the learning set by very many inactives. And so we use many thousands of inactives together with a small number of actives. We divide it into a training set and a test set. We produce the activity model with this iterative stochastic elimination engine. Once we have this activity model, we confirm it with a test set, and if it is confirmed, we can pass a huge set of molecules through this activity model, score all of them, arrange them, sort them from the best to the worst, pick the top molecules, and purchase and send them for wet experiment. So I'm not going to repeat. I've shown you this slide where we had already many research activities with this method. Those are molecules that we found and were recently patented as toll-like receptor antagonists in collaboration with Fraunhofer Institute. You may not see easily, but they are very diverse in terms of what we call the Tanimoto coefficient, which is the measure of similarity between molecules. They are all unsimilar, so they are very different from each other, and they are very different from the learning set. So we always find novel molecules that are active and also very diverse. And another recent success is the ISE discovery of iron chelators. In this case, we are trying to chelate ferric iron from ribonucleotide reductase. It's an idea for cancer studies. We have anti-targets, which we don't want to touch. We don't want to touch hemoglobin and myoglobin. We already found, when we screened 1.8 million molecules, we found 300 that could be active, but we chose the most 34 diverse structures, and 5 of them were shown already to be iron chelators of a new type that has never been seen before. I won't go more into the, I'll finish by this last story of multitargeting, which is a huge project that we started last year of screening all the GPCR, the main GPCRs in the central nervous system. And you may see that they are all interacting with each other, and there are lots of cross interactions which are not yet known in many cases. And what we are focused in the first case on the cannabinoids, because our school is specializing in cannabinoids. And we were interested especially in finding CB2 agonists that do not have any CB1 activity. And so we passed through this model. We passed 1.8 million molecules. We picked the top 67. We tested them on the models for agonist and antagonist of CB1 in order to avoid completely binding to that receptor. And so we purchased 35 molecules and sent them for testing in the NIH psychoactive drug screening program at the University of North Carolina, and we're waiting for the results. And I'd like to end by thanking our supporters, Fraunhofer Institute on the toll-like receptors, the Deutsche Forschungsgesellschaft on a project of BCR-ABL, TargetX in Hungary for selectivity to cancer cells, and the most recent one that I've shown you on day one, the Israel Office of the Chief Scientist of the Industry and Trade Ministry for the nanomupirocin, which is already advanced in advanced animal studies. And I'd like to thank my collaborators, the new ones that I've not shown you before, Professor Dom and Dr. Basu on the iron chelators, toll-like receptors. Studies have been done at the Fraunhofer Stuttgart by Professor Stefan Rupp and Dr. Anke Berger-Kentischer. The selective studies, the selectivity studies on prolyl oligopeptidase are done by Professor Sakaguchi, who did not send us his photos, so there you may see a very well-known photo from Japan. And this is my group. Shaima is responsible for the CNS studies, Dr. Noah Stern for the acetylcholinesterase, Ahuva Cern for the nanomupirocin, Maria for the toll-like receptor. Anwar Rayyan is, I'm supporting Anwar Rayyan's ideas, but he is the hidden source of many of the ideas that I've described before. And I'd like to thank my group and to thank you for your attention.